Welcome, everybody. My name is Megan Pistoles. I'm the Education Outreach Coordinator with the SLELO PRISM. I'll be facilitating today's webinar. This webinar is part of New York State's Invasive Species Awareness Week, also known as ISAW, a statewide educational campaign aimed to raise awareness of invasive species and encourage the public to take action to stop the spread of invasives. You can visit nyisaw.org to learn other ways you can participate in this campaign. I'd like to begin by just introducing the St. Lawrence Eastern Lake Ontario Partnership for Regional Invasive Species Management, or SLELO PRISM for short. It is one of 8 PRISMs that span the state of New York, creating a network of partnerships as an integrative approach to invasive species management. The PRISM network stemmed from recommendations of the New York State Invasive Species Task Force in 2005 and became fully established in 2013. The network is funded by the Environmental Protection Fund in coordination between the Department of Environmental Conservation and various host organizations. SLELO is hosted by the Nature Conservancy. Our region encompasses over 7,000 square miles of land in the counties of Oneida, Oswego, Jefferson, Lewis, and St. Lawrence, as well as Eastern Lake Ontario, the St. Lawrence River, and other water bodies within and bordering those counties. We collaborate with our partners to protect our lands and waters from the impacts of invasive species. Now I'd like to introduce our presenter, Rob Williams, the SLELO PRISM Manager, who will provide an introduction to how biological controls are tested and used effectively, discuss progress on controls for species such as water chestnut and knotweed, and share hands-on experience with using biocontrols to control and manage swallowwort. Thank you, Rob.
Thank you, Megan, and good morning, everyone. I hope everyone is enjoying New York State Invasive Species Awareness Week, and I'm hoping that everyone will have some opportunities this week to get outdoors and enjoy nature a little bit. Thank you for the opportunity to share this information with you today. Before I begin, I would like to figure out how to advance my slides, which are not advancing. There we go. I would like to acknowledge some people who are involved with biological control research, and these people have also been my mentors over the past few years. Lindsay Milbrath, Jeremy Biazzo, Carrie Brown Lima, Dylan Perry, Lisa Tewksbury, Bern Blossy, Wade Simmons, and Katrina Morgan. Again, these folks have been my mentors, and I have certainly learned a whole lot from these folks over the years. And before we get much further into this, I do want to say that biological controls can work, and they can be very effective, and they can offer long-term suppression of the target invasive species. You'll see on this slide here on the left, we have a pond with purple loosestrife along the riparian corridor on the left, and on the right, you'll see what that same area looks like about 5 years after the introduction of a biological control for purple loosestrife. It's a Gellerusella beetle. And you can compare this and easily, make the connection that biological controls can work and they can be very effective. That said, we've had to learn our lesson. We've had to learn, unfortunately, the hard way. And years ago, some of us may recall when the cane toad was introduced into the United States and other countries as a biological control to control the cane beetle. And at the time, there weren't a lot of regulations. If it sounded like a good idea, hey, let's do it. So that's how we did it. We brought in the cane toad to eat the cane beetles. They did a good job. But then the cane toad itself became highly invasive because there were no natural predators, and they also have a very high fecundity. So we introduced this biological control. It ate the target species, but at the same time, the toad itself became invasive. So that was a mistake. And I want to assure everyone on the call that that's no longer the case. I'd like to take just a minute to share with you the process today that we must undergo in order to get a biological control approved for release in the United States, and because we're connected ultimately in North America. And what you're seeing here is a very simplified version of the process. This comes from the Blossy lab down at Cornell. But it's important that we take note of how things work nowadays so that we don't make the same mistake that we made with the cane toad. So the first thing that you have to do is you have to identify a natural predator from the native range of the target invasive species. And then before you do anything, you have to have funding, because ultimately you're going to have to go to that region, wherever it is on the planet, collect some of these biological controls for research and bring it back into the US. And it takes money to do that. Once you've identified the natural predator, you travel, you obtain some specimens, then you have to get an import permit, which allows you to bring that species into the country and keep it in under quarantine and in a federally approved quarantine facility. There are likely others. The only quarantine facility that I am aware of in the Northeast is at Cornell University. So once you have the species in the quarantine facility, you begin your research. Research begins on what we call host specificity testing on a whole array of native species. And we'll talk a little bit more about that host specificity testing in a few moments. A lot of research takes place because you want to make sure that the species, the biocontrol, will not inadvertently go after non-target or native species here in the US. So you have it in the country, now you begin testing. Depending on how the research goes, you then have to petition USDA, the Technical Advisory Group, to begin what's called field trials. That petition is reviewed by our neighbors to the north and the south. And then once you have approval to do that, you'll receive a letter of concurrence from the US Fish and Wildlife Service. It goes through public comment period, posted in the Federal Register, etc. And then, if all goes well, there's a final ruling that takes place, which either allows you to conduct field trials or disallows you not to conduct the field trials. Once you have that authority and ability to do that, you then start what's called caged field trials, where you actually keep the biocontrol in a cage in the field. And depending on how that goes, ultimately you'll be able to liberate into a free living state. And the caged field trials are typically to address things like, will the biocontrol overwinter in this part of the country? How many bugs or insects do you need to control a given population of the target species? Things of that nature. We have to know these things. The whole process nowadays takes 15 plus years. It's very complex, and it's very focused and very specific. And the whole idea again here is to make sure that that biocontrol won't cause inadvertent damage to native species here in the US. So it's a very complex process nowadays, so that we can avoid making mistakes like we did with the cane toad. So I'd like to talk just a few minutes about several biological controls that are currently available or being studied here in the US. The first that I'd like to share with you is a biological control for swallowwort, Cyanantium species. In the SLELO region, swallowwort is perhaps one of the most aggressive terrestrial invasive species that I've ever encountered. Often we are considered the epicenter of swallowwort in North America. It is a very, very aggressive terrestrial species native to the Ukraine and surrounding areas. It has these long, slender vine and leaves with conspicuously pointed seed pods. This plant can produce approximately 2,000 seeds per square yard, and the seeds remain viable for approximately 2 years. A very aggressive reproducer, impacting nearly all the lands where it becomes established. So you can see a close-up of the swallowwort plant on the top slide, and kind of a field shot or a patch shot, if you will, on the bottom slide. And so there is a biological control that is currently in the caged field trial stage. It is known as Hypena opulenta. It is a moth in the family Eribidae, and its native range is from the same area as swallowwort, that being Ukraine and surrounding areas. The larvae feed and defoliate the leaves of the swallowwort plant, and once the stem gets about 50% defoliation, the stem dies. This is an approved biological control, and it's currently, as I mentioned, undergoing field trials here in New York State. We were able to assist with some of these field trials a few years back in 2018. We put a 6x6x6 cage in a shady site. We also put a 6x6x6 cage in a sunny site to compare what the habits might be in both shady versus sunny sites. And you can see on the screen here that the slide in the upper left is one of the cages in the shade site, and it's populated by pretty much all swallowwort in the cage. And then you can see the lower slide in the lower right-hand corner, where what has happened to the swallowwort about 6 to 8 weeks after the introduction of the Hypena opulenta larvae into that cage. What does that look like numerically? This is a slide showing the percent defoliation over time, and it compares the shade cage with the sun cage. And as you can see, the shade cage, in about 8 weeks, 7 to 8 weeks, there was a little bit over 80% defoliation. And the sun cage, during the same time period, there was a little over 30% defoliation. Megan, if you could mute everybody else, that would be great. So you can see that the sun cage, excuse me, the shade cage outperformed the sun cage, suggesting that the Hypena opulenta prefer or do better in shady sites. This is the same thing, only we're looking at the reduction in live stem count, and you can see again that the shade cage produced a very high reduction in stem counts, whereas the sun cage, although it did produce a reduction in stems, it was less than what we observed in the shade cage. So it does work. It works better in the shade, but nevertheless, it does work. So that's an example of a biological control that is currently in the field testing stage. Let me just go back to that again. We will be releasing additional bugs in several areas around New York State, hopefully this summer. And we will be working closely with the same folks that I accredited and mentioned as my mentors. So we're hoping to advance this biological control release in several areas around New York State. We do believe that the Hypena opulenta will overwinter, at least this far north. Our friends in Canada have had similar success with Hypena opulenta, so we're hoping that this one will help us to suppress swallowwort populations in the region. And I should also note, often is the case when we can't look at biological controls as a cure-all, where it will eradicate a particular target invasive species. More often than not, a biological control will help us to manage a target invasive species at a level of suppression. But it is an important tool in our toolbox, and they can be effective. There's another biological control currently being researched for water chestnut plants. It is a same genus insect as what was used on purple loosestrife, Gallirucella. This one is Gallirucella burmanica. And this, in the next several slides here are from the Wade Simmons down at the Glossy Lab at Cornell. So this is a beetle that produces multiple generations per year. It's a leaf-feeding beetle, and both the larvae and the adults feed on water chestnut foliage. This is just a slide of the quarantine facility down at Cornell, and part of the, I believe this is what the host specificity testing looks like, with different plants, native plants in each pail, and then you subject them to the potential biological control. So I mentioned that we talked just for a second about the host specificity testing. This is where it gets pretty complicated, but very important. And what the researchers do is they subject the biological control to various native species, and they don't give them a choice of what they can eat, and they just want to see if that biological control will feed on the native species when they have no other choice. They also do testing, which is multiple choice testing, where the biological control can feed on its preferred food, in this case water chestnut, but also you throw in a couple other native plants to see if they'll cross over and feed on that as well. And this helps the researchers to determine if there would be a non-target impact by bringing this biological control into the region. And I've heard that they have to subject the biocontrol anywhere from 40 to 60 or more native plants to make sure that they aren't going to cross over and start causing damage to native plants. So it's all part of the research and testing. This is the leaf damage that can be caused to water chestnut by the Gallirucella beetle. Again, the idea here is to defoliate the plant to a point where it cannot photosynthesize and therefore cannot produce seeds. Where are we in the process for the biocontrol for water chestnut? As I understand, the researchers at Cornell, Baron Blossy, hopes to submit a petition to the technical advisory group sometime this year, probably later this fall. And if they are successful in the remaining piece of the process, it's possible that they could see field trials perhaps next year if all goes well. Japanese knotweed. You can see a photo of the Japanese knotweed stems on the top photograph here. It is native to East Asia and the Koreas. It has large 6-inch by 3-inch, 3 to 4-inch leaves alternating on the stem, broadly oval and pointed with arcuate venation. And then the bottom shows what the Japanese knotweed looks like when it flowers and produces these white sprays. It is a very aggressive riparian invader. It can also thrive in wetlands, roadside drainage systems, and other hydric soils. It does spread primarily by rooting of plant fragments. So any piece of the plant that's 0.5 inches in diameter or more can sprout roots. The biological control that is currently being looked at is a psyllid from the same native range as the Japanese knotweed plant. And this particular psyllid, we are told, feeds on Japanese knotweed, but I should also note that researchers are suggesting that these psyllids are difficult to establish, and that the impact is light to moderate, to say the least. So let's not get our hopes up on this one, but let's look at it further to see if it can help us to suppress Japanese knotweed populations. This one, I believe, is ready for field trials this year. That's my current understanding. It will be a couple of years yet before we can determine the impact. It'll be a couple of years before we can determine the number of psyllids per stem you need to have a desired outcome. But I believe that's towards the end of the research process, and now we're into more of the field trial process. So fingers crossed on that one. I also wanted to share the fact that not all biological controls come from other areas on the planet. There is a milfoil weevil that is native to North America. It tends to prefer Eurasian milfoil to native milfoil. It's difficult, however, to sustain insect populations without augmentation. And the reason for that is, although these weevils feed on Eurasian milfoil, the weevils also make pretty good fish food. So there has been some work done in the past to augment populations and then introduce them to milfoil stands. You have to do that very carefully, otherwise the bugs simply, if you throw them in the water, they become fish food. But if you attach them to milfoil stems and then submerge the milfoil stems with the bugs into the water amongst an existing population of milfoil, they tend to have better survival rate. The problem with this is you need a lot of them to suppress the populations of milfoil, and there's no one that I know of that's currently raising these bugs and selling them or otherwise making them available. But they do exist. So again, the point here is not all biological controls have to come from the other side of the planet. And although we don't have time today, I would like to just share with you a few other biological controls that are either being researched or approved. There are several parasitoids that are currently approved for the emerald ash borer, and these are being liberated in several areas around New York State. There is another beetle and silverfly that has been approved to suppress hemlock woolly adelgid populations that are currently being released in areas of New York State. And then there's another biocontrol that is being researched for Phragmites, and it's a moth that comes from the same range as the non-native Phragmites, and that's currently being researched. Excuse me, at this point. The question and the concern with these are, if you find a biological control that is effective against the target species, you need to have some system of mass production. If you're going to raise these bugs and distribute them at a level that is needed to suppress the target species, then somewhere, somehow, you have to have a lot of these bugs, which requires mass production, and there's a cost to that. It's a lot of work by researchers, and it's just something that, you know, we're not really there yet in terms of, once we identify them, okay, now how do we mass produce them? So it's an ongoing concern. There is an update available on some of these biological controls that we've talked about. It's produced by the New York State Invasive Species Research Institute. I have copies of that. If you'd like to email me, I'll share my email address with you momentarily. I can forward that to you, or you can reach out to Carrie Brown Lima at the New York State Invasive Species Research Institute. I'm sure she would be happy to share them with you as well. That said, I could go on for probably the rest of the morning and talking about biological controls, but I think the point that I really wanted to share with folks today is that, you know, hopefully we can do prevention at its best and prevent the introduction of invasive species into our region. If we fail to prevent them, then hopefully our early detection and rapid response will pay off before a particular invasive species becomes well established. Once they become well established, all we can hope for is to suppress them or manage them below a level where we see a reduction in their negative impacts to ecology. So biological controls can be another tool in our toolbox. I think the biocontrol for purple loosestrife has done an exceptional job here in New York State in suppressing purple loosestrife populations. I see potential for the swallowwort biological control, the Hypena opulenta, to help us to somewhat suppress populations of swallowwort. And then some of these others that we've talked about, I think we're more in the let's wait and see how things go. But, you know, as I said, fingers crossed that we can add some of these tools into our toolbox to help us better manage biological controls. We've learned from our mistakes. We do things much better now. It is a very difficult, costly, and timely process to get a biological control approved. But once we get to that point, again, we can release these without concern over the impact to native species. So with that, I will close. And if you have any questions, I will do my best to answer them.
Thank you, Rob. We do have some questions in the chat box here. One question is, do the countries where the invasives come from, do they study biological controls for those invasives? And if so, do they share the information with us?
Yeah, so the answer to that is yes, absolutely. A lot of the researchers at Cornell and ESF and at USDA work with researchers in the other country. I know that for example, the biological control for water chestnut, the folks down at Cornell University, Baron Blasi, actually traveled to Southeast Asia several years ago to actually work with researchers at universities there to obtain some of the specimens and bring them back. And they share research and we share our findings as well. So the answer to that question is yes.
Excellent, thank you, Rob. And another question is, can you estimate when the Hypena opulenta may be widely available?
That's a really good question, and I'm hoping soon. We released some insects 2 summers ago, and they were released in various areas, caged releases around New York State. Unfortunately, that particular population was succumbed to a parasite that targeted the Hypena, and we lost most of the colonies here in New York State. Researchers both at Cornell and University of Rhode Island and SUNY ESF have been rearing bugs over the winter on a special diet so that we now, hopefully this summer, we will have more bugs that are healthy, and we'll be able to put those out into the field. We do need to do caged releases a little bit so that we can understand, you know, again, how many bugs it will take per stem to have an impact. Currently, if we have more than a certain number of moths, adult moths that appear in a cage, we do liberate them outside of the cages. But I would suspect that within 2 to 3 years, if the population and the colonies survive and do well, they will become more available year after year.
And then we've got another question. What do we know about the use of weevils for milfoil? How effective are they, and do they thrive after release?
Yeah, that's a tough one. My knowledge of the milfoil weevil is, yeah, they do munch on the terminal leader of the Eurasian milfoil plants, causing them to, be stunted in their growth, if you will. Again, when you put these small weevils in the water, it's tough to maintain a good population because, as I said, they make great fish food. So I don't have exact numbers for you. I've heard, folks say that they do work pretty well. I've heard folks say that they don't work because you can't sustain the high population that you need to see a noticeable difference in the milfoil population. I do understand that they are difficult to release into new areas because your release methods have to be such where you don't just dump them in and all the fish move in and eat them.
Excellent. Thank you, Rob. Are there any other questions? Okay, if there's no more questions, then that will conclude our webinar presentation for today. Thank you again for attending. There will be a recording of the webinar on our YouTube channel, so feel free to check that out. And we will be sending out a follow-up email with resources to those who registered for the webinar. Thank you for joining, and happy Invasive Species Awareness Week.